Hello, welcome back. Dr. Jenkins here. This is our chapters on the nervous system. So, there is so much we could cover, but we are going to try and just cover the main parts. As we're trying to cover the whole body in one semester, we can't get as detailed as I want, but that's okay. We're just going to cover the main parts, but it can still be a little tricky. So as always, make sure that you're spending enough time with the material and reaching out to me if you have questions. A cartoon to start things off. Hopefully this is not you. Ha ha ha. All right. So when we talk about the nervous system, there's actually many different parts to it. Simply put, there's really only two parts to the nervous system. The central nervous system, which is the brain, spinal cord, and the peripheral nervous system, which would be all the nervous tissue outside of the brain and spinal cord. Within both of these, central and peripheral nervous systems, there's nervous tissue that has a lot of important characteristics and that allows us to send nerve impulses. And then we're also going to talk about a branch of the peripheral nervous system called the autonomic nervous system. I know you've heard of that. That's the fight or flight, but it's really important. Okay, so before we get into those four parts, so we're going to talk about nervous tissue, then the central nervous system, then the peripheral nervous system, then we'll talk about a branch of the peripheral nervous system called the autonomic nervous system. But before we do that, we, of course, have to just kind of get our bearings, talk about the organization, and then make sure we cover all the functions of the nervous system. So, here we go, and this is what I already said, but let's just make sure we have it clearly and well. We have two components of the nervous system. Structurally, the nervous system is broken down into the central nervous system, which is the brain and spinal cord. That's it, brain, spinal cord. And the peripheral nervous system. Technically, the peripheral nervous system is everything, all the nervous tissue outside of the brain and spinal cord. But what that really comes to be is the nerves in your arms and legs. All right. Now, I love this chart. Bear with me here. So at the top, we have our two main parts, the central nervous system, brain and spinal cord, and the peripheral nervous system, which would be all the nervous tissue outside of the brain and spinal cord. Later, we're going to talk about some branches of the peripheral nervous system. So the peripheral nervous system has some organization of itself. But simply put, to begin things off here, we have two parts of the nervous system, central and peripheral. Right? Central nervous system and peripheral nervous system. Based on these two parts, we can give a function. So check it out. The primary function of the central nervous system. And remember, the central nervous system, brain and spinal cord. Well, of course, what does the brain do? It's our control center. So the functions of the central nervous system is really control. So this is our control center. Any processing. Think about our brain. It processes information. It allows us to make decisions, to send out nerve impulses to move muscles, right? So it is only in the central nervous system that we have the control center and the ability to process. Whereas in the peripheral nervous system, because it's only the nervous tissue outside of the brain and spinal cord, the only function of the peripheral nervous system, folks, is to transmit nerve impulses. I apologize, it's allergy season, so I'm sorry for blowing my nose there. So the only function of the nervous tissue outside of the brain and spinal cord is to send nerve impulses. That's a really important function, though. So our peripheral nerves send out nerve impulses from the brain and bring in nerve impulses to the brain. And then it's the brain and some parts of the spinal cord that are able to do the processing and therefore control things within our body. So I haven't seen that cartoon in a while. Can't someone else just do it? Of course Homer would say that. So we can see those functions in here. Our central nervous system, brain and spinal cord, control and processing. Our peripheral nervous system, which is all the nerves outside of the brain and spinal cord, communication. This is what we send nerve impulses, transmit nerve impulses. All right, so then it makes sense. Control center over the body, processing information, and then transmitting nerve impulses to and from the brain. These would be the three primary functions of the nervous system. We can put this into a visual form because this is how it always happens. You know, I believe it was way back in chapter 1. You know, remember when we first met? Haha. In chapter 1, we talked about homeostasis and a negative feedback loop. Well, really, the nervous system operates as a continuous set of nervous feedback loops. Really. So here we have sensory input from the body. This picture kind of cracks me up because, I mean, don't you think they could have picked something more interesting than, ooh, look at that glass of water. I really want some. But this could be anything. It could be that you want to pick up something from the ground. So you have to be able to see that and eventually use a muscle to pick it up. It could be an organ inside your body. Maybe this could be your stomach. I'm going to try and draw a stomach here. Not very well, but there's my stomach. So maybe your stomach is empty. So it's kind of like shrunken a little bit. And there are receptors in the wall of the stomach. So there's sensory input coming from the organs as well. Right? Actually, I have to correct this because, you know, from the stomach is the esophagus. There we go. So if the stomach is empty, there's less stretch, and those stretch receptors are sensory input. So all the sensory input is going to be coming into the brain via the spinal cord. So if we were going to break this down, this is the CNS, and then these two parts are the peripheral nervous system, and this is how they work together. It's a negative feedback loop. There's always sensory input from your eyes, ears, from your organs, traveling along peripheral nerves to your spinal cord, to your brain. Your brain processes the information. Oh, I really want that glass of water. How am I going to be able to pick it up? Or your brain processes, oh, the stomach wall is not stretched. It's empty. How am I going to tell the body to bring in food? As a result of that processing, then a nerve impulse comes down the spinal cord and then out into the peripheral nervous system to whatever organs or regions will do what your body needs or wants. So in this way, it kind of is like a negative feedback loop. There's some kind of stimulus, we go to the control center, and then we send out a motor response. This is saying the same thing. Okay, so we've covered the basic structure and the overall function of the nervous system. Let's start getting more specific. Let's talk about nervous tissue. Nervous tissue is really important. I mean, everyone's probably seen the neuron. This is the cell that sends nerve impulses. But there's also other helper cells around the neurons. Together, they make up nervous tissue, and this is how we're able to do the functions we need to do. So in this portion of the chapter, we're going to talk about the structure of the neurons and the helper cells. We're going to introduce something called the resting membrane potential, and I really like that discussion. Seriously, it's really good. And then we're going to talk about the basics of how your nervous tissue is able to generate an electrical impulse. That's really freaking cool. Okay, here we go. We have two parts of nervous tissue. We have the neurons. The neuron is what you think of. It's the kite-shaped cell body with one long extension called the axon. But what you may not think of are these other cells called the helper cells. More specifically, we'll call them neuroglia. So in this picture, we're seeing this here is the outline of a nerve cell, a neuron, right? So it would be kind of kite-shaped like this. That's the neuron. But everywhere around the neuron, I'll put it in red, we have these helper cells. Some helper cells look like mini neurons with extensions. Some helper cells will be like over here. There's all sorts of helper cells. I'm not going to ask you to know the specifics. If you're interested, there are many different types of helper cells. Astrocytes, Schwann cells, oligodendrocytes. But you just need to know them as a group. That in addition to our neuron, which has the kite-shaped body, we also have neuroglia, little helper cells all around. And actually, I'm not going to ask you this on a quiz or exam, but actually, by volume, we have more neuroglia, we have more helper cells than we do neurons. That's how important they are. Really cool. Okay. So here we go. This is actual nervous tissue. You see the neuron, and then all those smaller dots are neuroglia. They're everywhere. We couldn't have nervous tissue without it. It's a really cool picture. All right, make sure you review the basic structure of a neuron. This is the basic nerve cell. You know it. We have the cell bodies, kind of kite-shaped. We have dendrites. The dendrites are the many extensions coming off of the cell body. Dendrites. And then we have one long extension called the axon. So make sure you review those. What we're going to find is, functionally, it is the cell body that does the processing. What we're going to find, it is the cell body that does the processing. And because of that, the cell bodies are located in our brain and spinal cord. That's where we do the processing. Versus the axons. The axons are where we transmit or carry those nerve impulses. And the axons are found in the peripheral nervous system. Whereas most of the cell bodies are in the CNS, most of our axons are in the PNS. They're connected at the spinal cord, but it is the axons that are going to send those electrical impulses. And those are found mostly in the PNS, peripheral nervous system. Again, I apologize. All right. You may have heard of this before, but there's some really important characteristics. For example, covering the axon. So we've already established, coming off of the cell body, there's one long extension called an axon. Well, covering that axon are these little coverings. You see them? These are called, collectively known as, the myelin sheath. It's a little fatty covering over the axon. One of the most important functions of this myelin sheath is that it increases the speed of nerve impulse conduction. Without the myelin, the nerve impulse would travel slowly. Slowly. With the myelin, that nerve impulse goes boop. So it really increases the speed of nerve impulse conduction. Additionally, the myelin sheath provides some protection. I mean, nervous tissue is really prime real estate. In other words, it's really important. So our body wants to protect it. The axons do have some capacity to heal if they're damaged, but certainly not like other tissues in our body. Now, in between each of these individual myelin coverings, myelin sheath, is a space. And that space, those spaces, are known as the nodes of Ranvier. So the little gaps or spaces in between the myelin are called the nodes of Ranvier. Basic anatomy here. Now, you've probably heard of a synapse. Let's just make sure we understand what that is. We've already established a neuron, cell body, dendrites coming off of it, one axon. Now, when that axon comes to another nerve, they don't touch. There is a gap. Same thing here. There is a gap. And that gap between neurons, between the axon of one neuron and the cell body, or sometimes the axon of another, or dendrite of another, is called a synapse. What we're going to find out later is that we like that synapse because we're able to release chemicals. Maybe this one can release acetylcholine. Maybe in other times it'll release norepinephrine. These are called neurotransmitters. But the fact that there is a gap allows one neuron to release different neurotransmitters, which can have different effects on the next neuron. So even though it might seem inefficient, it really allows for a wide variety of communication. I can release different neurotransmitters to have a different effect on the next neuron. Look, what I just said. You know, I am telling you the truth here. I'm not making stuff up. So here we have, let's see, what color should I use? We'll do black. Here I have the axon of one neuron. This is the axon, leading into another neuron. This gap, this little teeny tiny gap, is called the synapse, right? So the end of one neuron doesn't touch the next thing. There's a little gap called a synapse. And what are we going to release across that synapse? Chemicals called neurotransmitters. These little green circles are storing neurotransmitters. And when the time is right, when a nerve impulse comes down, there's a series of steps that occur, resulting in that neurotransmitter being released. You can see the little green circle going up to the edge and kind of spewing out, almost like vomiting out the neurotransmitters. And those neurotransmitters cross the gap. And the benefit is that different neurotransmitters can deliver different messages. So make sure you know that the chemicals that cross the synapse are called neurotransmitters. And by the way, that makes sense. Nervous system, transmitter. We're talking about chemicals in the nervous system that transmit a message. And then also know that different neurotransmitters can deliver different messages. Good stuff, everyone. Good stuff. Okay. Now that we've covered the basic structure of a neuron, let's talk about how on earth do these neurons generate an electrical impulse? Because That's what a nerve impulse is, an electrical impulse. It's amazing. Now, to frame this conversation, we have to start with something called the resting membrane potential. Most people that I have in this class have not heard of this before. Maybe you have, but it's really important. And it's really cool, I think. But then again, I think everything is cool, right? Yeah. Science teachers, I tell you. So let's first talk about this resting membrane potential. To define the resting membrane potential, all it means is that at rest, cells have a negative charge on the inside of the cell. At rest, that's why it's called resting membrane potential, resting. At rest, there's this negative charge on the inside. And what this serves us as, it is a type of potential energy. You know, potential energy, it's like stored energy. It's not energy currently in motion, but it's stored energy. So when we want to use it, we can actually turn that potential energy into energy in motion. Really, really important here. Okay. Now, this is true of any cell. So it could be a muscle cell, a skin cell, a heart cell, a liver cell. It could be anything. We happen to be talking about nerve cells. So it's true everywhere. We're just talking about nerve cells. But the inside of the cell has a negative charge relative to the outside. That's really cool. Depending on where you are in the body, it could be -70, could be -90, could be -100 millivolts. In the neuron, the inside of the cell is -70 compared to the outside. It's true in the cell body. It's also true in the axon. So that negative charge on the inside continues negative on the inside relative to the outside at rest. At rest. Why do we have this? As I've already begun to talk about, because it is a source of potential energy. Now, when a cell is at rest, when a nerve cell is at rest, it's not doing anything. If I go back, this particular cell, it's at rest. It is not currently sending a nerve impulse. But yet, it has this negative charge, and that acts as a form of potential energy. And I give the example of batteries. If I just have these batteries sitting on the table, they're not doing anything. Yet, there's some chemicals on the inside that are a form of potential energy. Because, just like your nerve cell, just like the batteries, when I want to, I can take that potential energy and put it into a situation where I can get something. In this case, when I put these batteries into a flashlight, I have light. In the case of your nerve cell, when I want to, I can convert that potential energy into making a nerve impulse. If you started with nothing, in other words, what if the inside of the cell didn't have a negative charge? What if there was no potential energy? If I start with nothing, I've got no potential energy to work with. If you put something else in this flashlight instead of batteries, it's not going to do anything. So you have to start with something, this potential energy. So this resting membrane potential, the fact that cells have a negative charge on the inside relative to the outside, Even though at rest it's not doing anything, it's a form of potential energy. It gives us something to work with so that when we want to, we can take that potential energy and do something with it. In the case of a nerve cell, we take that potential energy and we make a nerve impulse. Think about a spark plug in a car. This is a better analogy for an electrical impulse or an electrical current. The spark plug at rest doesn't do anything, but when we trigger it, it's going to start the whole engine. And that's similar to what the nerve cell is going to do. Okay. Now, wouldn't you like to think that we're done? And I'm sorry to inform you that we're not, because we have to get into some more of the details. It's just not going to be good enough for us to just know that there's a negative charge on the inside at rest. We have to know how that gets there. And I'm not going to go over this just to torture you, even though I was saying this to another class I have. I'm not trying to torture you on purpose. There's a reason. So the reason why we have to know how this develops is because it's going to help us understand how that nerve cell makes a nerve impulse. Okay, so here we go. I've got a drawing on the right, but I'm also going to do another drawing. Let's say we have a nerve cell. There is our nerve cell. Cool. We do have a negative charge on the inside relative to the outside. And I want to make a point here. As you've noticed, I'm always putting that negative charge right by the membrane because that's where the difference in charge is. If you put a voltmeter to the center of a neuron, there would actually be no charge. So that negative charge is only going to occur around the membrane. So the basic answer to how we get this negative charge is because of ions. We talked about this in the chemistry chapter. Ions are charged particles. Particularly, we rely a lot on potassium and sodium. And there's many others, chloride, so many others, magnesium. I'm just talking about two, sodium and potassium. So let's talk about this. We tend to have a higher amount of potassium inside the cell. Excuse me. On the other hand, we tend to have more sodium outside of the cell. That's not to say that there's going to be some sodium inside, but there's always more sodium outside the cell. There's going to be some potassium on the outside, but there's always more potassium on the inside. So I would suggest that you start here. Again, I apologize. So work with me here step by step. You can absolutely do it. So we already talked about what the resting membrane potential is. Cell's got a negative charge at rest on the inside compared to the outside. And we are beginning to talk about how that develops. And the answer is an ion. So to start, we always have more potassium inside the cell, and we always have more sodium outside the cell. Okay. There we go. Always more potassium inside, always more sodium outside. Now, we're going to start to talk a little bit more because these ions will diffuse, and there's a difference in their rate of diffusion. So, I think it might be easier for me to start another screen here so I can draw it and keep going back to it. So let me just make a new one. New. Notebook. Create. I'm so sorry. Look, it didn't like me blow my nose, did it? There's a nervous system. There we go. Here's the untitled notebook. I don't like it either. Okay, so here we go. Let's lay out the scene. We have a nerve cell, and we have an axon. You know, I'll make it a little bigger. I have a tendency, my handwriting is small, so it's not always the best for showing things. So let's make it bigger. It's like hard for me to write it big. Okay. We know that. Let me see what colors I used. I used red for potassium. So here we go. We'll lay out the scene. We're going to have more potassium inside of the cell. And I use green for sodium. We're going to have more sodium outside the cell. Perfect. Okay. So, as is the case, when you have a concentration gradient and you have a semipermeable membrane, there will be diffusion. So, sodium will diffuse from higher concentrations outside the cell to lower concentrations in. Potassium will diffuse from higher amounts in to lower amounts out. Now, it just so happens that these ions tend to diffuse through protein channels. So scattered amongst our phospholipid bilayer are these protein channels. So what's going to happen? Potassium will diffuse from higher concentrations here to lower concentrations there. Sodium will diffuse from higher concentrations here to lower concentrations here. Fine. Yeah, we know about diffusion. Got it. Great. But these are both positively charged. Wait a minute. Sodium plus positively charged ion. Potassium plus positively charged ion. How are we going to get to that negative charge on the inside compared to the outside if they're both positively charged ions? And the answer is, simply put, potassium diffuses faster. Now, there's a lot of other things going on, but I'm trying to make this understandable. So I'm just saying there's a lot of other factors happening at the same time also. This happens to be one of the factors, and I think it makes my point well. Potassium diffuses out faster, a lot faster. I want to say the permeability of potassium is like 50 times greater than sodium. So when talking about this, I give an example. Let's say that every 1 millisecond, I'm just making these numbers up, but it will illustrate the point. What if every 1 millisecond, so per given unit of time, 10 potassium diffuse out? So the inside of the cell is losing 10 positively charged ions, right? Because potassium is leaving. So the inside of the cell is losing 10 positive ions. In that same unit of time, in that same millisecond, 1 millisecond, maybe only 3 sodium come in. So in that same 1 millisecond, the inside of the cell has lost 10 positive ions, but it's only brought back in, it's only gained 3 positive ions. So relative to each other, because potassium flows out so much faster than sodium can come in, what is the result? The result is, because potassium flows out so fast, we lose a whole lot of positive ions, and we can only bring in so many. The result is that the inside of the cell develops a negative charge relative to the outside. This is as simple as I can make it. There's other factors going on. There's other movement of ions and a whole bunch of stuff. But the point is, we lose the inside of the cell loses so much potassium, and it can only bring in some sodium, that the inside becomes negative relative to the outside. Now, we're getting into some physiology here. Your exams are multiple choice, so I'm not going to ask you to write an essay, but I absolutely will ask you questions about this in multiple choice. So I am expecting you to have a basic understanding here. Let me just peek ahead for 1 minute. Okay. Now, there's other things going on. We're going to have, you may have heard of this, we're going to have another channel somewhere else around the cell, which is called the sodium-potassium pump. So there are a bunch of other things going on here, but in an effort to simplify, I'm not including everything. I just wanted to give you an idea that ions are what's important here. Ions, the movement of ions, is what is creating this difference in charge. Now, why do we care? Well, you as an EMT or paramedic, that's what most of you, I believe, are taking this course for, you might see this. When someone is dehydrated, not only do they lose water from their blood, they also lose sodium. You know this. So what's happening in dehydration is, instead of being able to have 3 sodium ions chilling out here, when we're dehydrated, we lose water from the extracellular space, but we may also lose sodium. So if I'm dehydrated, maybe instead of being able to bring in 3 sodium, I don't have as much sodium, so I can only bring in 1. And if I can only bring in 1, what happens to the resting charge? It actually gets greater. So when we have dehydration and we alter the amount and flow of these ions, you are messing up the resting membrane potential. And guess what? If I'm messing up the resting membrane potential, and I'm messing up or throwing off the balance of ions, I'm also going to maybe affect the nerve cell's ability to make a nerve impulse. I mean, what are some of the symptoms of severe dehydration? Confusion, headache, disorientation. So when you are severely dehydrated, you lose sodium in addition to water through sweat. You don't replace it. We throw off this balance of potassium and sodium. It messes with this resting membrane potential, and it messes with our ability to make a nerve impulse. That's why there are nervous system symptoms. So this will come up. So when someone's dehydrated, we not only want to replace them with water, we need to properly replace the electrolytes so we can maintain this proper balance of ions between the sides of the cell membrane. Wow. I'm imagining everyone out there just having all these wonderful realizations. Like, wow, 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 I get it. So great, wonderful. Teach me more. Is that how you're feeling? Listen, I'll just take it if you're awake. I'll just take it if you're listening to this. You're already a step ahead. All right. So now that we understand what the resting membrane potential is, we understand how we get there by the flow of ions. Let's take it to the next step. Now let's say we want to use that potential energy and no longer have the nerve cell at rest, and we want to make a nerve impulse. Here we go. Okay, first of all, a nerve impulse is an electrical current that travels down the axon. The scientific word for a nerve impulse is an action potential. So you might see me use action potential or shorthand it with AP. But an action potential really is just a nerve impulse, an electrical current that travels down the axon. we're going to cover 3 steps to tell us how the cells make this electrical impulse, how they make the nerve impulse or action potential. Okay, let's see. So let's go back to this, right? We talked about that. Now, we know how things work at rest. Let's lay it out. And I think once we get through this nervous tissue, we'll end the first video, and then we'll pick up a second video for the rest. So here we have rest. We know at rest, nerve cell, negative charge on the inside, positive charge on the outside, and just to throw a number out there, the inside of the cell of a nerve is about negative 70 millivolts. Rest. So here are 3 steps, boom, 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 for how we make a nerve impulse, starting at rest. So from rest, what's the first thing that has to happen? Something called depolarization. Depolarization just means that the sodium gates open. We open more of those sodium channels. So let's lay it out. Step number 1, depolarization. What's going to happen? Cell body. And this can happen along the cell body, but it also happens along the axon, right? So just like the cell body, the axon is negative on the inside, positive on the outside. That's rest. When we depolarize, we get there by opening a bunch of sodium channels, and it causes the inside of the cell to now become positive. So we already know we have some channels, right? So we have sodium. Sodium diffuses in. Potassium diffuses out. Great, got it. Right. Well, what happens if we suddenly open a ton, not really a ton, but we open a lot of sodium channels? So in addition to just that one, now we open more. So what's happening with the ions? We have a sudden influx of sodium. If you all of a sudden have a bunch of sodium, and remember the sodium's coming in here too. When we have a sudden influx of sodium, now what was negative 70 on the inside of the cell, we just flood it with positive ions. Well, now the inside of the cell becomes positive, and the outside becomes negative. So we flip-flop the charges, right? So where at rest, the inside of the cell is negative. When we depolarize the membranes and we have a flood of positive sodium coming in, that negative charge on the inside suddenly becomes positive. It will usually go all the way up to positive 30. I'm not going to ask you these specific numbers, but I think that it's good to just mention them. Because think about it. If, in the matter of a millisecond, maybe even less, if, in the matter of an instant, a cell goes from negative 70 to positive 30, that is a big change in voltage. Now, it's not big in electrical outlets in your home millivoltage or voltage, but in the body, that's enough electricity, let me tell you. So think about it, negative 70 to positive 30. How many millivolts change is that? Negative 70, negative 60, negative 50, negative 40, negative 30, negative 20, negative 10, 0, positive 10, positive 20, positive 30. That is 100 millivolt change. So simply by opening sodium channels and having a flood of sodium come in, we generate, so we take that potential energy and now we've generated 100 millivolt change. And that switch from -70 to +30, that 100 millivolt change is a spark. Just like a spark plug. Beep, spark. That is what depolarization is. Then that spark spreads down the axon. So, step number 2, spread of depolarization. So, where we initially opened sodium channels in one region and we had a spark, let me put it in black here. Let's say we started by opening the sodium channels here and we had a spark. Well, now we open sodium channels here and we have a spark. Then we open sodium channels here and we have a spark. So a continual spark. Spark, spark, spark. That is what the current is. That spread of depolarization generates a current. Spark, 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 spark. So that's how we keep that current going. We open sodium channels in one place, then we open them in the next place, open them in the next place. And that depolarization wave will spread. Now, after an area depolarizes, it will repolarize, which just means going back to what it was at rest. If we changed it by opening sodium channels, wouldn't we want to bring it back to normal by closing them? Yep. So step number 3 is repolarization. Those very same sodium channels that we opened, now we close. So now sodium stays out here. There'll still be a little bit of diffusion, but now we keep all that sodium out there relative to the potassium in here. And if there's no longer sodium rushing in, the inside of the cell will return to being negative because potassium will diffuse out faster than sodium. So after an area depolarizes, and that depolarization wave spreads to depolarize the next, which causes a depolarization wave to spread to depolarize the rest. After it depolarizes, sodium channels close and it repolarizes. Inside goes back to -70. These are the basic steps. How this works is, and don't laugh at me, do I have another example here? No, okay. The best example I can think of would be the wave at a sporting event. So if you imagine the wave, okay, here's our dudes, right? In the wave, one person stands. That is like depolarization. When he stands, doesn't that cause the next person to stand? And when the next person and that causes the next person to stand? That's the spread of the depolarization wave. But immediately after someone stands, which causes the next person to stand, this person sits back down. So depolarization would be the same as standing. The spread of depolarization would be the same as when you stand, the person next to you stands, which makes the person next to them stand. And then repolarization would mean after you stand, you sit down, back to normal. And if you can watch the wave going around a stadium, you can imagine it's one line. It's one impulse going all over. It's like you can follow the wave. Oh, look, now the wave's in the outfield stands. Now it's in the first base stands. Now it's behind the plate stands. That's what a nerve impulse is like. Because that depolarization wave spreads, spark, 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 it can move down the axon. The movement of this spark down the axon is called propagation. Fancy word. And the reason why nerve impulses can travel so quickly is because of that myelin sheath. When you have the myelin sheath, the impulse doesn't have to spark at every individual one. When you have the myelin sheath, you only have to spark here and then here. So the spark kind of jumps, making it go faster. All right. Okay. Just literally a couple odds and ends, one odd and end. That's it. And then we'll stop this video before we begin our next one. Each nerve impulse, each action potential is the same intensity. This is important. When we depolarize the membrane and go from -70 to +30, it always depolarizes at that intensity. It's always a 100 millivolt change. So each spark is the same intensity. But you may ask, well, can I have different intensities of nerve impulses? And overall, the answer is yes. You know, we talked about the nervous system responding to stimulus. If you touch something really hot, you accidentally touch the stove, you're going to pull away really quickly. And that's possible because we're sending 100 nerve impulses really quickly. So your body is basically responding. It senses, oh my God, stove is really hot. And it sends 100 nerve impulses, like a machine gun fire. Impulse, 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 impulse. And that's how you could quickly pull away. Versus if I touch something that feels a little bit sharp, but not really, my nerve impulse response is different. Oh, I guess it's kind of sharp. Maybe I guess I'll pull it back. So even though each individual nerve impulse is the same intensity, we can change the overall intensity by sending more nerve impulses. Like I said in that example of touching a hot stove, if we want to send a really strong signal, we just send many more nerve impulses back to back to back to back to back. Because that's how it works. We're sending nerve impulses all the time. So even though each individual one is the same intensity, if I want a big overall effect, I'll send 100 really quickly. Versus if I don't need as much of an overall effect, maybe I only send 5. All right. Good stuff, folks. Spend time with that. I'll see you in the next video where we'll start talking about the central nervous system.